afternoon, everyone. What an honor and a huge responsibility being here in my native Azorian Islands, presenting some of my most exciting work, the work that I like to talk about, really motivates me and drives me. I would start with a really short introduction of myself, if the slide would move forward. Oh, there you go. So I'm basically a native Azorian marine biologist. I was born here in the middle of the Atlantic in one of the Azorian Islands. I guess I didn't really have a choice. When I was about 6 years old, like my daughter Teresa here, first time I put my goggles in the water and I saw the first glimpse of the underwater world, I guess that wired my brain in a certain way that I basically was almost inevitable that I would end up here. Not here specifically, but at this stage of my life and doing what I do. And the Azores Islands, this is basically my backyard. This is where I do most of my research. Really, this is a very special place where you have this meeting of these three tectonic plates where they meet the Gulf Stream. So this really highlights the uniqueness of this ecosystem, and where a lot of the megafauna and the marine apex predators meet. And don't just take my word for it. Here's this picture is from a paper that we published recently, and you can see here how many different marine megafauna and apex predators meet in the Azores and then disperse from the Azores at some point in their lives. You have marine mammals, you have fish, tunas, billfishes, sharks, marine birds. This is a contribution of many of our colleagues that work here with us. And so this is really, the Azores is really a hot spot in the middle of the North Atlantic. But I was asked to talk about marine apex predators. I didn't really suggest this title was not mine, so this is kind of a disclaimer. I'm going to do my best. And I don't want to be very extensive about the definition of what is an apex predator, because it's not very straightforward. But the general idea is that sorry, it's just too early. A marine apex predator is an animal that occupies the top of the food chain, and they control their prey, and they lack natural predators. Like the mako shark. The short-fin mako shark is a good example of an apex predator, but what about when they're born? So it depends on their life stage. So basically, even the great whites, when they're born, or an orca, a killer whale, they probably are in the menu of some of the other predators. So you're not always a top predator. What about the large filter feeders like these whale sharks, the massive whale sharks that aggregate here around Santa Maria Island in some years, and they feed in the same prey as these big, massive bluefin tuna and yellowfin tuna. They share the same prey. They don't have natural predators because they're huge. They're almost impenetrable. Are they apex predators as well? I think they can be. What about the mesopelagic or the mesopredators from from ecosystems that have been depleted of their large predators? So now they are the largest predators. These guys would normally not be the top predators in a healthy ecosystem, but if you remove the big predators, then they become the big predators, then they are apex predators in a specific context, right? But even the whale sharks, they fear, because they have even other top predators that are more, let's say, vicious. They're capable of actually attacking a 4-meter, 5-meter long great white shark and prey on them. This is the case of the killer whales that can actually, sorry, this is going too fast. That is actually, they actually target the great whites, and they actually are afraid. They've been shown to be sort of afraid, and they avoid these areas where even though there's plenty of food, they avoid these areas where the killer whales are, and they avoid those areas even though there's a lot of forage food there. And then what about the greatest apex predator of all, humans, that are able to prey, all the apex predators. We've removed most of the apex predators. We've changed the way ecosystems look. We've altered most of the ecosystems on the planet by removing the apex predators. Sorry. This picture is from the 70s, and it was taken just around the corner here in San Miguel Island. In the 70s. It's a great white, when there used to be plenty of great whites roaming across the Atlantic Ocean. So it's puzzling, right? It's not straightforward, the definition of an apex predator, right? But I just wanted to sort of clear this, and so we'll be on the same page, and it's good to have this for context. But you're probably asking, why should we care about the apex predators? Well, by now I think it's pretty clear that the apex, for most of us, that apex predators, especially sharks, that control the food webs, they balance the food webs, they keep the reefs healthy by controlling the prey, which in turn are very important to keep the systems healthy, to keep especially the coral reefs, and not just coral reefs, other reefs as well. In the case, this is a very good example of how the overexploitation or removal of top predators, this is a well-documented case in the scallop fisheries, multi-million-dollar scallop fisheries in the US that basically collapsed because as the top predators, the sharks, were removed, the cow-nose rays exploded, their population exploded, which resulted in the depletion of the scallops that are their favorite food. And so this resulted in the collapse of the multi-million-dollar industry with all these social economic impacts that come along with that. On the other hand, most of these top predators, or apex predators, they are also deep divers. And what happens is they are very important in connecting and allowing the flow of energy and the cycling of carbon between the deep sea and the top layers of the open ocean. Also, we take advantage of the apex predators, like the previous presentation, that by deploying data loggers and cameras and other equipment on these top predators and in megafauna in general, they can allow us to understand much better the functioning of our changing oceans. However, in the past 50 years, we have explored down the food chains and we have removed much of the biomass, about 70% of the biomass of sharks and rays, due to an increase of about an 18-fold in fishing pressure around our oceans. This is a map of a recent publication for a large consortium where we also take part. And this shows basically how overwhelming the industrial longline fisheries is in terms of occupying almost the entire ocean. And basically what this shows is there's very, very little refuge left for these top predators, for these apex predators. We think that what this work shows is that 75%, 76% of all of the oceans overlap, of the ships fishing in long lines in the ocean overlap with populations. Of the 16 species of sharks populations that we've studied on this paper, they overlap about 76%. There's very, very little refuge left. These apex predators, these pelagic animals, are incredibly diverse, and there's still many knowledge gaps that preclude us from understanding or designing effective protection and management measures, because we don't know a lot about. It's difficult to study these animals. We don't know where they're going for the most part, why they're going, how they are moving, and why they're moving. So biologging is a great tool to understand these aspects of their ecology and help us and assist in designing better, more effective conservation and management measures. But biologging has evolved greatly, is a great tool really, but the attachment methods have lagged behind the great advances in biologging and the technology that we use. For the most part, we need to fish the animals, restrain them. We change their behavior as we do this. So if you're very interested in a very specific behavior locally, that's not going to work. And so that motivated me to think about an alternative, a non-invasive alternative to attach and instrument these animals. And so basically the idea, I usually say, had to come from outside the box. And so the idea came one day. I was freediving in a seamount where mobulas aggregate, and I saw these mobula stuck on an anchor line, and she couldn't move back. She was stuck there. I had to go there and release her so she could keep going about its life. And so the idea came about this, to design this lasso that would be, like you see here, the first time it was done, it was filmed. it goes around the head and it's retained on the shoulder, then it trails a tag behind. This was just a very simple tag made on the shop just to test the concept, and it works. It was stoked. And then we tried it on blue sharks as well, and again, it also worked. And we were very excited also because we kept all the fingers in our hands after we were doing this, and now we do this all the time, and we still have 10 fingers each. So that's pretty cool. And this allows us to get a really cool view of what's going on in the lives of these marine megafauna, and we having no impact. We've demonstrated that these methods have very, very low impact on their behavior, and so we're very happy with that. And now we've been deploying these tags on multiple animals across the globe. But then we wanted more. We wanted really specific tags, and our list was very long. We wanted tags that had cameras, that had red lights, that could measure behavior, that had accelerometers, magnetometers, had GPS. So we had to go and partner with our colleagues from engineering at Ceia, mainland Portugal, and we designed this, one of the tags that they've designed for us, and that we've been deploying. This is an example of a deployment on a Chilean devil ray that we have here around the Azores, two of the only known aggregations in the world for this species. And they work really, really well. You can see the animals, they're not changing their behavior. They stick around once we deploy the camera tags, or even the multi-sensor tag like this one. We've deployed it on whale sharks, we've deployed it on hammerheads, on tiger sharks. So we're really happy with this because it's allowed us to understand a lot better the lives of these animals. This is an example of the first deployments we did on a Chilean devil ray. We found that they can dive as deep as 2,000 meters, which totally changed the way we look at these animals. We thought they were surface dwellers, they like warm water, but they can be down at 4 degrees Celsius, 2,000 meters. Pressure is overwhelming, there's no light, and they can do it. That was so amazing when we discovered this. That was really changed the way we look at them. The other thing we found is that they are incredibly powered, great divers. They do these power and glide dives. They go so fast that the camera stalls and goes into spin and control until it slows down, and then the camera stabilizes again. And this is, again, we thought they were just slow surface dwellers. They don't like to speed up too much. But then when we look at the data after seeing that, what we can see is that you can see those on this graph, basically, you can see the descent, the red line. What they do is when they start descending, they go really, really fast, high amplitude movements. They speed up really fast as the fastest animals on the planet, or fishes on the planet. And then when they get to halfway through the descent, they start gliding. And they basically glide, same speed, but they don't use any energy. That was so surprising. And we think that may explain why they have these rete mirabilis, which is this network of thin blood vessels that connect the muscle and the brain. And we think they're producing heat to then warm the brain as they are down there at 4 degrees Celsius at almost 2,000 meters depth. Also, looking at the accelerometry, we look at the way they come up from the dives, and it's really convoluted. We had no idea. When we look at the dives from a 2D perspective, we just see it going down, going up. Then we start looking at 3D, and it's like they're going in a corkscrew movement up and down, or up, going up, actually. And this is really intriguing. We think this might have to do with foraging and looking for food strategy. Also, we can see that the way I mentioned earlier, the way they recycle carbon, this is the big poop of a devil ray that's being filmed by the devil ray that's coming behind. So you can help us quantify how much of the carbon they're recycling, for example. We can see how much of the fish they feed. They're not feeding on plankton when they're here because there's no plankton. There's really oligotrophic, so they're feeding on fish. They're hunting with other fish. We can see, for example, these animals that look pregnant. The first time that we saw this, what looks like to be the pup kicking inside the left uterus of a pregnant female, which might suggest that the Azores might be an important pupping ground. We don't know where they pup anywhere in the world. This could be one of the places. This is another interesting view of one of the apex predators that not always predators. They also take the opportunity to scavenge on a dead sperm whale if they have the opportunity. It's a free meal. The ocean is really big and it's hard to find. People are easier to find these days. This is the same perspective of that shark. They found a group of shark watchers in the middle of the ocean. It's a very unusual view. I would say. And we also did some work with reef mantas in Hawaii, and we found this. For the first time, we documented this incredible behavior with onboard sensors and cameras. I mean, it speaks for itself. This was truly incredible and amazing, and we were so lucky because the camera detached right when it went back in the water at the moment that it was supposed to detach. We also documented the incredible relationship between tunas and the massive whale sharks that aggregate here in the Azores, and we found out that this relationship is what allows these massive whale sharks to feed on fish, because again, there's no plankton here in the summer. So what they do is the tunas gather the bait balls, and then the whale sharks go and they can attack the bait ball and swallow the whole bait ball at once. And then when the tunas were gone after a storm, and I was in the submarine, and this is a shark with a camera that we had deployed a couple of hours before, we were hanging on a marine reserve seamount, and all the jacks were around the submarine. And what the whale sharks were doing, they were coming in, they were gathering the tunas and taking them away as if they were tunas. So basically to replace the tuna and do the work for them to gather the bait balls, and so they could feed, they could continue to feed as the tunas were gone. Lastly, well, just 2 more slides. Just want to talk to you about very recent data that we got from the short-fin mako shark, which is a really understudied species. We don't know anything about the way they move, the way they behave, the way they dive. And much like the cheetahs on Earth, which are the fastest animals on Earth, the mako shark is the fastest shark on the ocean. But they don't, like cheetahs, they don't run, they don't sprint all day long because it's a lot of energy. It's very, very demanding. So what they do, you can see the red dots basically is the peaks of burst speeds, right? And so what they do, they swim slow like most other sharks. The big difference is they don't glide. And I think the reason why they don't glide is because they're not built for gliding. Much like a jet fighter, which has short wings, they're built for speed. That's why it's called the short fin mako. So it has short pectoral fins, doesn't give them that much lift, but it's great for speeding, but it's not great for gliding. That's why they don't glide, I think. And plus, during the night, we also found that they stay much more calm during the night. They don't do those sprints, those bursts of speed, etc., suggesting that they're not as active during the night. The last one I want to show you is this data from the GPS tags, multi-sensor tags that we deployed on to study the blue shark diving and shamming operation, because people were concerned about whether or not doing this close to the islands could represent some sort of risk for the people on the beach. It would be the sharks would be competing with the same resources as the fishermen because they would get used and stay around. But what we found is that every time that you remove the chum, all the sharks that we've tagged, they go offshore. They go into the island slope. That's their habitat. That's where they like to be. So we helped basically understand and give some hard data for a profitable debate about whether or not there is risk associated with. And so the last slide is about what's next? What's the next frontier? I'm really excited now because we live in an era where the technology is available like never before. The miniature sensors, they're so cheap, they're easy to access, and now we're working on new tags that will allow us to understand better the physiology of these animals that are also deep divers, that move across oxygen gradients. We want to learn how their heart rate changes or deals with this intense pressure and differences in water oxygen, how the oxygen in their blood varies, how they control their temperature, because for example, the blue shark is not an endotherm, so it doesn't have any control. Apparently, it doesn't have any means to control its internal temperature, but it does dive into really cold water. So we want to understand how they manage to keep the body warmer than the external water, than the outside water. We want to understand how their DNA is fixed when they go into these extreme, really demanding environments of deep water, cold water, low oxygen, etc. And finally, I would just like to ask you all for your attention and apologize because I'm 4 minutes overdue. And also, of course, thank all the people that have been, this is collaborative work, of course. Many people contributed to this, and also the funders, of course, which are also key to develop any kind of this work. So thank you very much, and I hope to talk to you soon again.